All right, so first I want to thank the organizers for giving me an upgrade from the 1-minute presentation to a short talk. I'm going to redact the second part of my title today for the sake of timing and coherence. If you want to hear about the meiotic recombination story, which is very interesting, I'll have a poster, I think it's number 24. So in my laboratory, we're interested in understanding the molecular basis for a number of pathologies that arise in a variety of rare human diseases that are associated with defective DNA damage responses and chromosomal instability. And generally speaking, these fall into 4 major classes. You have increased cancer initiation and progression, infertility, different indices of premature aging, and severe developmental disorders that affect primarily the central nervous system and the immune system. And so depending on the disease, you'll have different combinations of these pathologies. But among the most poorly understood and perhaps most debilitating in some cases are the central nervous system disorders that are primarily characterized by microcephaly or neurodegeneration. And one example of this is Seckel syndrome. This is a Seckel syndrome patient with a mutation in ATR. And these patients are characterized by microcephaly and sometimes very severe dwarfism, although there's a pretty wide spectrum of presentations. There's 8 known loci in humans. Not all the genes have yet been identified. But interestingly, these genes fall into 2 major classes: genes that are involved in the DNA damage response, particularly responding to DNA double-strand breaks, and genes that function at the centrosome. Not all the functions are known, but they localize primarily there. In some cases, have very well-characterized functions. And this is just a picture of a mouse model from Oscar Fernandez Capetillo's group, showing that, at least in some cases, we can recapitulate some of the phenotypes or pathologies that arise in humans. But there's very few models of these, and we don't really understand whether there's some sort of unified underlying mechanism or whether there's much diversity. And so we're very interested in the identification of the CEP63 protein as a target of the DNA damage response. And the only thing at the time that was known about this protein, this is work from Vincenzo Costando's group in 2009, was that the protein localized the centrosome and it was 63 kilodaltons. But they found it was a target of the ATM and ATR kinases. And they shared with us unpublished data from his student Nicola Brown that it interacted with the CEP152 protein. And so given that all of the known interactors with CEP63 are involved in neurodevelopmental disorders, we considered that this was a very good candidate gene for Seckel syndrome or related diseases. And so we set out to generate a mouse model of this. We got a gene trap allele that reduces the expression to undetectable levels. Whoop. As we first got our mice and our first MEFs in culture, our hypothesis was confirmed by the group of Fanny Gergley and Jeffrey Woods in Cambridge, and identified 3 female human patients with mutations in CEP63 and a mild form of Seckel syndrome, not as severe as what was seen in ATR. And they also had identified the CEP152 interaction. And work from both of our groups collectively showed that a major cellular function of CEP63 is to recruit CEP152 to centrosomes to promote central duplication. And if you knock down or knock out CEP63, you have a central loss phenotype, mild chromosome instability, more pronounced ploidy defects. In the case of DT40, where Fanny was working, very severe mitotic defects or mild mitotic defects, we didn't really see major mitotic problems in fibroblasts. So this was a bit unclear how important this was. And so we've, for the last few years, been studying our mice, trying to understand if this is a useful model for understanding a little bit about the etiology of particularly the CEP63 Seckel syndrome. So our mice recapitulate the major pathologies in humans. They're runted, so they have growth defects, and they have microcephaly. And at birth, the size difference is not so much, and the overall brain size is quite similar with the exception of the cerebral cortex. This is a major target in most of these diseases, and you have a thinner cortex. And so we were interested in understanding the mechanism. And the 2 major proposals that have been put forth to explain microcephaly both revolve around depletion of the stem and progenitor population. What's clear in some cases is that you have a defect in the regulation between symmetric and asymmetric division, so you favor differentiation, thus exhausting your stem cell population. You can't finish creating the structures you need to in the brain. In other cases, it's been proposed that chromosomal instability arising from mitotic defects or DNA repair defects leads to cell death by activating P53 or other pathways. And I don't have time to go into the data, but we examined the ability of these cells to self-renew in culture. There may be a minor defect, but this doesn't seem to be a major problem. However, we see very pronounced cell death in the developing embryos. So we look at embryonic day 12.5 or 14.5. You can see increase in cleaved caspase staining. So in all cases, I'm showing you here the cortex with the ventricle at the bottom. So the stem and progenitor population is here, and the more differentiated cells above. We compared this also to a very well-characterized DNA repair mutant, ligase 4, which is more severe. These actually are embryonic lethal, whereas our mice actually have a normal survival despite the microcephaly. And you can also see here the increase with tunnel. So we wanted to characterize what the cause of this cell death was, as we assumed that this was probably the major driver for the smaller cortex size. And so we looked first at the recruitment of CEP152, as this was the major function of CEP63 that we and others had found in cell culture. And just to show you an example of this, in U2OS cells, if you siRNA deplete CEP63, we can't detect CEP152 localizing the centrosomes. The protein is there, we can IP it, do a variety of things, but you impair its localization. And this gives you problems in centrosome duplication, although the overall phenotypes for the cells are quite mild. So we looked in the brain, so again, looking in the cortex, and the centrosomes are clustered along the apical membrane, so you can see very strong gamma tubulin staining here. In the wild types, you can see, again, CEP152. In our mutants, we can't detect CEP152 localizing the centrosome. Again, we can IP the same amounts of protein, so this is a localization defect. And so we asked, now focusing on this population, this is where the mitotic cells should be found in a normal brain. What is the behavior of these cells? Can we see signs of DNA damage or repair defects or other issues maybe related to centrosome function? And so we did a careful analysis of the stem cell compartment, and we were very surprised, given, as I said, the very mild phenotypes we see in cell culture. Nearly 50% of the mitotic cells form monopolar spindles. So this is the stem and progenitor population along the ventricle. And if we focus on just the metaphase and anaphase cells, we can see that many of them, approximately half of them, don't have a detectable gamma tubulin focus at one of the poles. So we have acentrosomal spindles and monopolar spindles. And the consequence of this is going to be tetraploid cells. We looked at markers of DNA breaks, and when we see a mild increase in gamma H2AX, we don't think that DNA damage is a major driver of this. However, we see a massive P53 induction in the ventricular zone where the stem and progenitor population is proliferating. And we did the genetic experiment, and we can completely rescue brain size back to normal by knocking out P53, and this is also responsible for all the DNA damage that we're seeing. Now, we also crossed these mice with CHEK2 and ATM. These are kinases that rescue many of the microcephaly models based on DNA damage, because after breaks, they're required to activate P53. And here we see no rescue, again, pointing more for a centrosome-based mechanism. So our model is that recruitment of CEP152 by CEP63 is, for a reason we still don't understand, critically important in the stem and progenitor population in developing brain. Loss of CEP63 function gives you centrosome loss, monopolar spindles, and drives microcephaly through P53. And this is genetically and mechanistically distinct from other models that we characterize, such as the ATR mutants and the MCPH1 mutants. And we're now trying to understand what activates P53. Tetraploidy and acentrosomal spindles have both been proposed. The mechanisms are not understood. And what are the consequences of this long-term in terms of tumor genesis or brain function? And I'll just say, we see the only other major phenotype we see is similar to the brain, very small testicles, but this is for a completely different reason. It's not stem cell based and not due to mitotic problems. I'd be happy to tell you about it on the poster. So this is mainly work from a postdoc, very talented postdoc, Marko Marjanovic, who was shared between my lab and Jens Luters. He had a lot of help from Carlos and more recently Berta, who did much of the brain imaging I showed you. Also contributions from Philip and Suvi in my lab. These are collaborators and funding agencies. And we have another poster on chromatin regulating kinases by Sandra Diakon. Thank you.
Thank you.